Hello, my name is Shinori Chowdhury, and I'm doing a presentation on the effect of methylglyoxal on E. coli YQHD and ADPH dependent aldehyde reductase. Whew. So, we're working with E. coli. E. coli we're working with strain K12, and it's easily grown. It's gram-negative bacteria, and it's very well known. It's been experimented on many times in history. So, the reason why we're using E. coli, like extra information, is because it's easily grown, yeah. We can find out the differences between the normal E. coli and E. coli in the presence of something else, which is methylglyoxal. So we can compare those two very easily, which is why we're using E. coli. So methylglyoxal is the phytochemical we're using to, to treat E. coli with. It's very highly reactive. It's it creates oxidative stress on these cells because if you look in the figure, you can see like two oxygens bound by double bonds on on a carbon chain, three carbon chain. It's a known phytochemical and it's like toxic to cells. The reason why it's toxic is because it reacts with like RNA, DNA, proteins, anything it can get its hands on basically, and that will like create problems such as bad genome, destroyed genome, uh, not properly transcribed RNA, which will lead to like not properly made proteins. Okay, so the genome we're working with is called YQHD. It's react it's a genome that transcribes the RNA, which makes a protein coenzyme NADPH dependent aldehyde reductase. So it's the blueprints for that enzyme, and what we want to see if that increases or it stays the same in the presence of methylglyoxal, which leads to my hypothesis. If what I think is that if E. coli was grown in the presence of methylglyoxal, its transcription of YQHD and the amount of coenzyme NADPH would increase. The reason why I think that is because methylglyoxal is very toxic. It needs to fight. The cell needs to find a way to survive against that. So that's why it would increase, uh, like measures to fight against it. So a procedure, bacterial growth. We grew two separate bacterial LB product cultures, one in the presence of methylglyoxal and one without. And the methylglyoxal positive broth, we add the phytochemical during the exponential phase. And the reason why we did that is because if we add the methylglyoxal in the lag phase or the stationary phase, for the lag phase, the cells won't even grow that well or it just might all die out due to the toxicity of methylglyoxal. And for the stationary phase, the cells are already towards death, so there wouldn't really be in like any changes there or any noticeable changes that we can calculate or measure. Nope. So for our bacterial harvest, we used a forced centrifuge at 12,000 G for 2 minutes. We took the supernatant out and used wash buffer to rehydrolyze it and centrifuged it again. Took the supernatant out again, and then we froze the pellet at negative 80 degrees Celsius for our next experiments. So for the enzyme assay, we didn't actually do the enzyme assay. We didn't have time due to the virus. So we took the cell pellet. Well, most of the cell pellet, hydrolyzed them in heat beast buffer. The reason why I did that is to like make sure that the proteins don't get denatured and keep them in the right pH. We sonicate them in 15 to 30-second intervals. The sonication, what it does, it ruptures the cell membranes for its contents to come out. The reason why we sonicate them to 15 to 30-second intervals is so that we don't heat up the solution too much. It would denature the proteins. And then after that, we centrifuged it at 12,000 times G in room temperature, and we kept the supernatant. So this the pellet were just the cell membranes and whole cells that we just didn't need to work with. And for And in the supernatant, we added the lysis buffer with protease inhibitor and glycerol. So the reason for that is to keep the proteins intact with the protease inhibitor and glycerol. Okay, so for our enzyme assay, we had to use a Bradford assay for a standard curve to figure out how much enzyme there was in our lysate. And then after that, we had to find out the optimal amount of enzyme needed for reaction. And then we did the reaction to calculate enzyme activity. As you can see in the schematic, the methylglyoxal turns into acetyl with the help of NADPH that turns into NADP positive, and that's the alkyl reductase. YQHD could be that. That's what we're banking on. Okay, so procedure, RNA extraction. So we used the GeneJet to extract RNA. We just followed the protocol for it. And the basic idea is to use silicone membrane and alcohol to catch the precipitation of RNA, and to use wash buffers to elute the RNA out of the silicone membrane, so we can use it to uh, so we can use DNase to cleanse the RNA from any DNA contamination. Because if there's any DNA contamination in our RNA, that would mess up our RT-qPCR experiment. Because if there's any DNA in our experiment, then that means there would be like any false amplicon races in the threshold versus our DNA ace free, which would be like no false information if it's done correctly. After that, after we purify our RNA from any DNA, we want to use reverse transcriptase so we can turn that RNA into DNA. We use the second method, which is just RNA one script and 2 times reaction mix. So what this does is that there's RNA, there's the one script, which is the reverse transcriptase, and in the reaction mix, there's the buffer to keep like everything nice and make the reaction go smoothly. And there is also RNase, so the RNA will be eaten up after it's turned into cDNA. We just followed the manufacturer's protocol. So for RT-qPCR, we use CyberGreen that shows us when the application starts increasing exponentially. So the more ampcon there is, the more fluorescence there are, as you can see in the graph. And when the graph starts to go up, that's when the threshold reaches, and that's when we can say that okay, this is ampcon in the uh, and the enzyme reaction and the yeah. So if the thresh if the threshold cycle reaches earlier, that means there's more amplicon in the reaction. And if the threshold cycle reaches later, that means there's less amplicon in the reaction. This would tell us that if there's more mRNA, let's say, in the reaction, then the threshold cycle would start happening sooner. If there's less, then it would happen later. Okay, for NPN assay, we had to add NPN with EPS buffer and cells. There were 6 different controls. So each category control would be a no methylglyoxal, and no cells at all. And for each of those, there'll be NPN or no NPN. We calculate the fluorescence for everything, and then no, we had to measure the fluorescence for everything, and then subtract the NPN positive with the NPN negative, and then subtract that with the no control to find out our NPN factor. So our results are the RT-qPCR was not good. So the idea is the lower the CQ, which is the threshold cycle, the more mRNA was transcribed. For RT, this makes sense because, well, there's only like DNA. For RT negative, it would be higher since reverse transcriptase was not like it's RT negative. There's no reverse transcriptase. And for blank, there are no RNA. So our blank, our CQ was pretty high, which makes no sense. For RT negative, for MG positive control, the RT is also high, which also makes no sense. So for our YQHD primer, we can't exactly accept this experiment. We have to redo it. For top B, it makes uh, kind of sense. But even then, our Mg is greater than Mg negative, so which means that uh, there was more alpha in Mg negative than there are in Mg positive. So for our enzyme assay, we use the backup results that we got from our lab technician. So our Mg negative was slightly higher than our control, which is like by about 5 units per milligram. And the reason why I think this is because there's other things in the lysate other than coenzyme and ADPH dependent reductase. So it's still greater, but not by a huge amount, but it's still significant because of the reason I just said. Alright, for our enzyme assay, we also did not do this experiment. This is a backup. Um, our NPN factor for control was 5.2, and our factor for methylglyoxal was 7.9, which means that methylglyoxal um, had a role in the increase of permeability of the cell membranes. This is due to the high reactivity of methylglyoxal. It probably reacted with the membranes, which allowed the membranes to be more permeable. And then more methylglyoxal got into the cell, which would react with everything else. E. coli would have to find a way to counteract this, and it does have genes to combat methylglyoxal, and one of them could be YQHD, which we try to measure in our lab. So this leads to our conclusion. The qPCR experiment was inconclusive. We just have to repeat the experiment again properly. Our enzyme assay gave a slight increase of activity for Ng control, which we found out. And then Mg is proven to increase membrane permeability. So using this information, we can conclude that methylglyoxal increases membrane permeability, which allows more methylglyoxal into the cell. This would force the cell to transcribe enzymes such as YQHD, which we don't know yet, is inconclusive, which would create more enzymes to turn methylglyoxal into a less reactive compound by reduction. There might be other genes that are transcribed to combat methylglyoxal. YQHD could not be the gene to do that. But we did prove that E. coli has ways to combat methylglyoxal in its own genes. These are my references, and thank you for watching my presentation. Bye-bye.